Hello, everyone. Thank you so much for joining us for a special look at what our restoration teams are doing to help restore and protect the deep coral habitats of the Gulf of Mexico. My name is Erin Spencer, and I am the Communications Manager for the Mesophotic and Deep Benthic Communities projects, working to restore the deep seafloor habitats in the Gulf that were injured by the 2010 Deepwater Horizon oil spill. Now, I know that is all a mouthful, so we are going to kick things off in just a minute by talking through what some of that means and what those habitats look like in the deeper parts of the Gulf and why some of them have needed our help. Then we were going to talk to our friends involved in the coral restoration work and get an inside look at a lab that is furthering what we understand about these incredible mesophotic and deep benthic corals. Now, our original plan for this was to actually come from live from the RV Point Sur, which is a 135-foot ship that was out in the Gulf of Mexico. But unfortunately, they had to come in early to avoid this incredibly large and dangerous storm that is currently in the Gulf of Mexico. Our thoughts are with everyone impacted by that major storm in the Gulf, and we're thinking about them and all of our partners and folks affected at this time. So, of course, as part of fieldwork, plans can change. And so we will not be coming to you live from the Gulf, but we have some really incredible footage and behind the scenes look of what happens to some of these corals once they come from the Gulf and into the labs. And we have some wonderful experts here joining us. As we go along, please share any questions or comments that you have in the chat, and I'll make sure that those are asked. And we will pause for questions throughout, so feel free to jump in when you have them. And in just a few minutes, we'll connect with the folks in the lab. But for now, I'm going to go through a little bit of background of the project and why we are doing this work. So as you travel deeper into the ocean, less and less sunlight is able to penetrate the water. If you go just past the recreational scuba diving limits, you reach something called the mesophotic zone, which means middle light. Despite the low levels of sunlight, this zone is home to colorful corals, sponges, fish, and invertebrates. Beyond this twilight zone, there is no sunlight at all, and it gets much colder and it is under much higher pressure. But there are still vibrant habitats full of life at these depths, hundreds to thousands of feet below the surface. These mesophotic and deep benthic or seafloor ecosystems are found worldwide, including in the Gulf of Mexico. And you can see some shots that we got here from these gorgeous ecosystems in the Gulf. All this footage was taken there. However, the 2010 Deepwater Horizon oil spill dealt a heavy blow to the Gulf of Mexico communities on land and at sea. 11 people lost their lives, and natural resources, wildlife, and habitats across the northern Gulf were severely impacted. More than 770 square miles of deep sea habitat were impacted by the spill. And as a result, collaborative restoration projects are underway to help us understand and restore these seafloor ecosystems found below 160 feet. These projects are developing new and improved maps of the seafloor to locate these important coral communities, learning more about the species that live in these habitats, the environmental conditions they thrive in, and how they might be connected across the Gulf, determining the best methods to encourage deep sea coral growth and reproduction to help restore impacted coral species and the important habitat they create, and connecting the habitats and restoration work to the public and natural resource managers. This restoration work is made possible by the Deepwater Horizon Natural Resource Damage Assessment Open Ocean Trustees, is led by the National Oceanic and Atmospheric Administration and the Department of Interior, and is joined by many partners, including a large number of academic institutions and non-governmental organizations. And if you want to learn more, you can scan this QR code and read all sorts of additional information about this work. So for the work that we're going to focus on today, we just concluded this expedition in the Gulf with the RV Point Sur, and that expedition was part of a series that have taken place since 2021 that focus on collecting coral samples for husbandry, reproduction, and genetic studies, as well as surveying mesophotic areas to find large coral communities and collect information about the surrounding environments. This project and sampling expedition in particular was in search of coral diversity that adds to a series of work by sampling and surveying unexplored areas, collecting live corals for new studies on health and growth, which we're going to talk a little bit about today, and filling sampling gaps, looking at different types of diversity in those benthic habitats. Now, I know that was a lot of background information, and so now we're going to get to the fun live stuff where we're going to go live from the NOAA Coral Lab in Galveston and be joined by one of our experts on the MDBC team, Chris Gardner. So let's see if we can connect to them now. Oh, good. All right. So here we are live from the Coral Lab in Galveston. We're getting an inside look at where some of these corals are being studied. And then throughout, Chris is here to join us and give us some great background on some of this work. So first things first, Chris, would you mind introducing yourself and tell us a little bit about your role on this project?
All right, thanks, Aaron. Like Aaron mentioned, my name is Chris Gardner. I'm a research fishery biologist, and while I work closely with the Galveston lab, I'm based in Florida in the Panama City NOAA Fisheries Lab. And I'm one of 3 project managers and technical advisors that oversee a lot of the experimentation and really management of the coral propagation techniques project. So some of my expertise lies within offshore sampling, experimental design, and whatnot. So corals are definitely a new and exciting post for myself, which is spent most of my career with fish, but this is exciting, and really have to give a hats off to Ben and Shannon and Bill in the Galveston lab. Really do a fantastic job taking care of the critters there.
And we're going to get into more details about this, but can you give us kind of an overall picture of how the work of the coral propagation techniques team contributes to the restoration of coral habitats in the Gulf?
Yeah, absolutely. So a lot of these, mesophotic and deep corals are very slow-growing, long-lived species, and we really don't have a lot of information about them. Things as simple as what modes of reproduction they use. Apologies, there's dogs and kids running around today. But with these studies, we're able to go in and try to figure out how to best restore the areas that were damaged by anthropogenic events, such as the Deepwater Horizon oil spill. So when the corals were damaged, often we were trying to replace those with new corals, where either we're going to be doing coral gardening, where we'll take a snip of one healthy coral and then outplant it to a damaged area, or like in the lab where they're let's see, spawning new corals and let's see, getting ready to outplant those guys.
And so you're talking about outplanting and all of this really specific work that is important to the restoration. For those who might not be as familiar, we're throwing around a lot of words like coral propagation, et cetera, et cetera. Can you tell us about what kind of process is involved with coral propagation and actually define what that means for us?
Yeah, Aaron. So for propagation, what we're trying to do is think of it like coral gardening. We're outplanting these things. We're like little bonsai trees. We're trimming. We're trying to create more coral. So when you trim, you can stimulate growth as well as move that material to another colony. So where it will grow into new material. So the goal with all of this is to propagate or put out more material than was lost in some of the injuries.
And we do, we see some examples here of this was happening in the field. And Sasha in the lab, hi Sasha, our colleague Sasha, was just showing us a couple of different corals in the same tank, and some look smaller than others. So those small corals are going to grow into those larger colonies. Am I right?
Yeah, absolutely. So, these are, individual polyp colonies. So they're a colonial organism. So they will, reproduce and grow into a larger colony. And once it gets up to a little bit larger size, where we don't worry so much about its survival, all these little baby corals. So these are actually Thesia, and the small purple ones are actually born in the lab. So they're spawned eggs. Their eggs were fertilized, settled out onto tiles after a few weeks, and I believe these are approaching their second birthday right now. So you can see they're really, really slow growing, but once we get it up to maybe 3 or 4 inches, that's when we think that their survival is really going to be there's a much better chance they're going to make it out in the wild.
And only being 2 years old and still being that small really emphasizes what you were saying about how slow growing they are. And can you tell us what these little tiles are for that we're seeing the corals on?
Yeah, so these are called settlement tiles. They're used often in the aquarium trade. We have several different materials, either ceramics or kind of like a concrete mixture. And basically, they're just used so we can, once the fertilized eggs will settle down, they metamorphosize into a little like worm-like stage where there's kind of a little grub thing crawling around, and they'll find nooks and crannies, and they will kind of, plant their foot down there and say, okay, here's where I'm going to live and grow up. So from then, we can take those tiles and move them, and once they're ready to be outplanted, we can put those without damaging once again.
And here on the video, we can see some sediment tiles that are going actually back into the field.
Yeah, so these tiles are these are trying to measure natural settlement, or recruitment of corals. So these larger tiles, we're trying to mimic some of the areas on the seafloor. You can see a colleague at University of Rhode Island, Jack Gerard, made these, and he's implanted all kinds of rocks and stones and things to try to mimic the nooks and crannies, because if they're on just a plain surface, they're probably going to get eaten by a sea urchin or a small fish or something. But with the recruitment, we're trying to find out under normal, conditions, how many of these critters are going to be, in an area. And that's just some of the basic biology questions that we really don't have an idea of in these mesophotic environments, mainly because it's quite difficult to work out here. You see these divers, and these are very specialized divers that require a large ship and recompression chambers and years of training.
And, for those that, since we're talking about some of the field work, for those that may have not seen that before, can you tell us a little bit what types of field techniques that we use in order to get those corals into the lab for study?
Yeah, absolutely. There are two main ways that we capture corals. Now, you saw the technical rebreather divers a moment ago, and we can use those divers, and when they go in the water at the shallowest depths for the most mesophotic corals, they can get between 20 and 30 minutes of bottom time. And then after being on the bottom for 20, 25 minutes, they'll spend the next several hours decompressing. So, to spend 20 minutes on the bottom, you might spend 3 hours in the water. So they can go and grab some critters pretty quickly. And they're going to the larger colonies and basically taking gardening shears, and we'll just snip off, a branch, for lack of a better word, of these corals, and then, transport up live, in Ziploc bags, basically. And then they're housed and brought to the aquarium. The other method that we use would be remotely operated vehicles. So we'll use underwater robot with one or two basically claw arms that it's like very much this is a very fancy one right here, with a 7-function manipulator, which is every ROV operator's dream, that can move pretty much every way a human arm can, and some ways it can't. But use that to basically just with a razor knife and cut a piece of the coral off, go drop it in a basket, and then bring it up, submerged in water the whole time.
And you mentioned earlier about there's so much about this kind of basic biology of these mesophotic and deep corals that we just don't know. And so even just a few samples can teach us so much about the biology and ecology of these animals. And once these corals make it to the lab, a lot of us don't have any experience of what it means to actually care for a coral in a lab setting. So can you talk us through a little bit about what sort of care do these corals need once they are in the lab, out of the field?
Absolutely. Yeah. So, if anyone has ever kept an aquarium, it can be difficult. If you've kept a saltwater aquarium, it can be very difficult. And then corals are just kind of the next level of difficulty. These these critters live in areas that are generally, very steady. They have, low temperature fluctuations. The deeper you go, the steadier the things are. And they're filter feeders also. So they require quite a bit of food to be input into the system. Okay, here on the screen, you see one of our species, Swiftia exertia, and it's being fed brine shrimp or sea monkeys. And you can see they're basically, the polyps are like lots of little sea anemones. So they'll grab, pull them in, and digest those. Now, the trick is for caring for these guys, they demand very high water quality. And so it's a delicate balance to feed them enough to have them grow and be healthy. And if you look at these corals, I mean, these guys are doing just a stellar job. And you can see how dense the polyps are. They're open, they're happy. These guys look as good as they do in the wild. So I'm super impressed. But there's a lot of water quality. You have to change the water frequently. There's there's a whole building there full of equipment. Water filters, sterilizers, protein skimmers, and these guys are constantly monitoring things. They have computerized controllers where they're looking for water parameters to get out of whack, as well as doing manual testing pretty much every day to make sure everything is correct with the water chemistry.
And it looks like we are watching these corals be fed live as we speak from the Galveston lab.
Yeah, so he's target feeding, with a little eyedroppers and go and put the food mixture, and not sure exactly what he's feeding today. I can't tell if those are live brine shrimp or there could be a mixture of some other specialty coral foods. But a lot of this work, these guys are on the cutting edge. They're working with a group of various aquarium partners, and through a lot of trial and error, a lot of work has been done over the years with shallow corals, but really, for the mesophotic and deep, it's definitely kind of a brave new world, and trying to take some of those techniques and then adapt them to deeper waters.
This is amazing, and I love that we get this. This is a really rare view of getting to see this happen in real time. So really, really excited that we're here. A reminder, I have plenty more questions, but as a reminder, if you have any questions and you're watching us live, please feel free to put it in the chat. But as folks are thinking of their questions, my next one for you, Chris, is, we talk about getting them in the lab, but can you talk about some of the questions that we're trying to answer with this lab work?
Yeah, absolutely, Aaron. So, when we're trying to restore these things, and that's, putting more out on the bottom than we took, we need a lot of, there's a lot of questions, and we need studies to inform restoration. So we're looking at, one of the most basic ones is how do they reproduce? So, they can reproduce in several ways. You can fragment them where if one's cut, it will start growing somewhere else. They can also reproduce sexually, where they'll release, their gametes into the water. And then, after a period of time, the eggs will fertilize and settle. And each one of these species, seems a little differently. Okay, there's video right now of a Swiftia, and you see the little tiny eggs being released, which is just super exciting that they're not only able to do this, but they're able to collect these and rear them, have quite high survival rates for years. So it's really a testament to what these guys are doing. So other studies we're trying to do. So aside from just basic biology, we're trying to figure out how to maximize their growth. We want to, make sure these things are healthy and that we can get them up, and with the ultimate goal of getting them back in the water. So there's a vial of fertilized eggs right now, right there, and I believe someone told me earlier there were about 1,000 larvae in that jar. So those will eventually turn into little baby corals, and by taking these fertilized eggs and raising them in an environment where they're not going to get all eaten, right away, and hopefully increase their survivability, we can get them up to a larger size where they can be put out and have a good chance of surviving.
And if I understand correctly, these were from coral samples that were just taken on the expedition last week.
Yes. So these were collected last week offshore of the North Florida, Alabama area, and so think right offshore of Pensacola, Mobile, just right there. And these things are literally right under our noses, sometimes pretty close to shore.
And just so remarkable at how quickly this restoration work, how much we're understanding about these corals, and it's just such a special view to know that this is a little glimpse into life at those depths. So can you talk to us a little bit about the sensitivity of these corals in their environment? What have we learned about their resilience in this work in the face of threats?
Yeah, so there's kind of a double-edged sword. So on one hand, these things are incredibly fragile. They don't do well with chemical changes to the water, temperature changes, anything like that, especially being oiled or having other chemicals placed on them. On the other hand, mechanically they're much tougher than they look. You'll see them oftentimes, going, swaying back and forth in the current. They're filter feeders, and they live. We're trying to figure out how old these guys are right now, currently, but certainly many decades, if not hundreds of years. Some of the deep water species can live for thousands of years, and just being able to recover, having an impact injury of something running into it is definitely pretty impressive. You can see some of the movement in those guys. There is a skeleton underneath the tissue and polyps, so it does provide some rigidity.
Yeah, and this is something I think, especially for those folks that might be familiar with only more shallow water corals, is really just so mind-blowing to see the structure of these corals that live in such deep and cold and or chilly and dark depths. We do have a question from one of the viewers.
Okay.
Is there any interest in assessing coral microbiomes while they are in activity?
That is an excellent question, and yes, that's some of the studies we're doing are within the microbiome. We have colleagues with INCOS and NOAA, Coastal Ocean something. They'll kill me for not remembering their acronym. As well as the USGS. We work with Chris Kellogg down there, and she's looking at the microbiome. Tom Grieg and others, NOAA, looking at how the microbiome, one, at their time of entry. So we'll take a sample right when the coral comes up, and then at various time periods in the lab, see how things change, if at all. So I know some of those data are forthcoming. So I don't have the results as of yet, but yes, there's quite a bit of work going on with the microbiome. It's a very exciting field.
So keep an eye out in the literature, is what I'm hearing.
Yes.
We have another question as well about if there's any proposed or active monitoring programs for those corals that are outplanted.
Yes. So currently we've outplanted corals on a few different expeditions. Some like in situ or ex situ, where we'll collect the ROV, fragment them out, and then place them in racks back down. Earlier this summer, we did monitoring of corals that were down right at 1 year, and they looked fantastic. So we had a couple different sites, and there were a few different experimental treatments of substrate type they were put on and whatnot. And there was some declines in areas that were a little more silty, but the areas basically where the corals were thriving, they're really doing well. They had noticeable, tissue gain, growth, and so again, those results are also forthcoming. But yes, we're definitely monitoring those. And oh, okay, there's a rack of that. From the looks of that one, that's one of the racks that was been out. I'm not sure if that's taken a year or just prior to a year, but we'll go back from the measurements of those, and you can see they look healthy and happy. And a lot of them, you can see growth down at the bottom of the bases. Without seeing the intake picture, it can be difficult to judge exactly how much they've grown. But if you see tissue growing down over the base, that's a good indication.
What a cool view to see that as they're back in the habitat. We have a couple more questions. What is the deepest depth that you've collected corals? Do you know that off the top of your head, the deepest?
Off the top of my head, within a few meters. Yeah. So I believe some of the deepwater corals, just over 1,000 meters, maybe 1,080 or so.
It's pretty deep.
And yeah, it's very deep. And okay, hang on, I have to pause from that question for a second, because they're showing, they're in the lab, Ben and Sasha are showing Mauricia larvae. And so these are larvae from those bright white corals. And, I believe these were just recently in the Galveston lab, they had the first spawn of these ever in captivity that I know of. And so that's
so cool.
It is so cool. But yeah, so back to after that quick break, back to the depth. We like to collect corals a little bit shallower, because it seems to be something about around that 1,000-meter threshold that they have difficulties deeper than that. Now, we've tried replicating pretty much everything we can, cold water tanks, dark, but seems to be a pressure component of one of the proteins in the coral that helps them digest food, that kind of unfolds. So we still have to figure that out before we can go super deep, if we do.
So that actually applies to one of the other questions that came up, which was asking, does the lab need to replicate the pressure that these deep sea corals were collected in?
Yeah, that's a great question. Thus far, we have not. With the mesophotic, with the ones, they're a little more robust, coming up from 100 meters or less. And now for human, that's still a ton of pressure. But these guys don't have any voids or gas pockets, so you don't have quite the pressure squeeze as you would, say, if you're diving down and you feel pressure in your sinuses and whatnot. But we do know that is a factor for some of the super deep ones with their proteins.
And now we have another question as well. And this is switching gears just a little bit, but this is a really important one because this comes up all the time. So someone asked, what degree would be the best for a career working in coral? I was thinking of going for environmental science, but would I need a degree in marine biology?
That's a great question. And there are a lot of different paths to get to this point. I always advise people to, regardless of the degree, get in and do a internship or a volunteer program. There's a lot of places around, depending on your locality. But that's what I did many moons ago, and that kind of steered me in the direction towards fisheries. So I don't know. I'd say that a lot of the times the experience is more important than the actual degree. I don't know, Aaron, you might have different advice on that?
Yeah, what I always tell folks is I took a very traditional, path with going on to graduate school and doing marine biology. But there, you can apply really any skill or interest that you have to working in ocean conservation. There's a lot of, for example, nonprofits that need very interdisciplinary groups of skills that maybe if you're outside of working of just the field work or the lab work. So I would agree with you, though, for sure, Chris, that getting some hands-on experience is really great if you can do that. But the other thing I would say is if you can't get hands-on experience, maybe like an undergrad, definitely don't count yourself out. There's a lot of folks that do work in other things and then move into the marine conservation space and the restoration space as well.
Yeah, any science degree also that concentrates lots of sciences, maths are incredibly important, statistics.
Yeah, as much as some folks try
I hate to say that. I struggled with math forever, and I started doing statistics, and it made sense. But
we are getting a little low on time, and I want to make sure we touch on this last question, which really, if you could for us just sum up how this coral work is really helping the restoration goals in the Gulf. And then as part of that, sometimes working in this field can be emotionally draining. There's a lot of issues facing the Gulf and our ocean. Can you also speak to what gives you hope throughout all of this?
Yeah. So for the first part of the question on, let's see how this is impacting the restoration. So out of the MESFOC and Deep Benthic Communities project or projects, whole suite of projects, this is the most direct for restoration, where we're going to be putting these corals and outplanting them back in areas that were documented to be damaged. So we have some pretty discrete locations where we, under the oil stick, where we had camera surveys out and saw these corals, and there are quite a few different species. However, there's about 4 species that accounted for over 70% of the injury. So if we kind of narrow down on those, and that's Swiftia, Thesia, Mauricia, and then in the deep sea, Paramauricia, those we get a lot of bang for our buck. And so that's what we're trying to focus our efforts. And, these things just really tie into the whole ecosystem. If you're a fisherman, you might look at this and see habitat, and, which is quite important. I mean, it supports quite a bit. And then thinking to the second part of the question with what gives me hope, looking at these videos right now, this is from an expedition earlier this summer. We were on a technical diving crew trip, and seeing some of the collaborators, it really came from all over the world, and seeing people just pour their heart into this, working so hard. We were easily working, 18-hour days every day for weeks, and people volunteering their time to do so and doing some really pretty intense things, diving hundreds of feet deep and just working like crazy and just ecstatic about it. And it's nice to see that, and really just see how excited the younger generations are, as well as the older. We show people these, like, wait a minute, that's alive, that's an animal, that's right in our backyard. That's really pretty special.
Well, I think that's a wonderful way to sum it up. And I want to say a huge thank you, Chris, for sharing your expertise with us. And thank you, Sasha, behind the camera in the Galveston Coral Lab. Really grateful that we were able to see such a cool inside view of this incredible work. And thank you to all of you who have joined us online. Really, really appreciate your thoughtful questions. Now, if you enjoyed today's event, please feel free to share this. We actually have a series of live streams throughout our expedition season, and this is the last one of the season. So thank you to everyone who has helped make these a success, and all of our partners and everyone who has joined. You can see all of the previous live streams on the links that are being provided in the chat. And we will also have a whole nother set of wonderful live streams coming to you next year. So make sure you follow along and stay tuned for those. To learn more about these projects, we're also sharing a link in a chat that you can read all about the different work, not just the coral propagation work, but the other projects that are happening to restore the Gulf. Also, if you wouldn't mind, we'd really appreciate if you could complete a short feedback form, which is going to be in the chat. And that's a way for us to make sure we are making these programs as great as possible and as useful as possible. But again, thank you all so much for your time, for joining us from all over the place. And we really appreciate it and hope to see you next year for our next series of live streams.
Hey, James.